Assalamu alaikum. On behalf of Ahsan Academy, let me welcome you to a presentation on connective tissue. This presentation will include the definition of the connective tissue, general characteristics and function of this tissue, composition of this tissue, features of the cells present here, and the constituents of extracellular components. Connective tissue is one of the four basic tissues of the body, and it is widely dispersed basic tissue. It forms the framework of each organ and also the framework of the whole body. You can see the two microscopic slide. The black arrow on the left figure is indicating the connective tissue, and the middle figure, this is the black arrow, is indicating the area filled with connective tissue. You can see the figure, the left figure, the upper part is the epithelium and below is the connective tissue. It predominantly comprises of extracellular matrix. You can see the epithelial tissue packed with cell, and there is minimum intercellular substance, but below that you can see the cells are widely dispersed and is mostly filled with extracellular matrix. The cells are of different types. You see there's a spindle shape, there are rounded cells, so different types of cells are there and widely separated from each other by the extracellular matrix. The arrow is indicating the connective tissue. The right one is a spread preparation of the connective tissue. You can see this is the microscopic structure. The cell columns are there, and in between cells the arrowhead is indicating the area of the connective tissue, and that area contains blood vessels. In the right figure, the arrow is indicating the connective tissue, and it is forming the framework in which the actual cells that is functioning, that is known as parenchyma. So parenchyma is situated within the framework of connective tissue. In general, it forms the stroma. Already I have mentioned. Each solid organ comprises of stroma and parenchyma. Parenchyma is the main functioning cell, and stroma supports the parenchymal cells and give environment so that this parenchymal cell can function. This whole body framework is formed by the connective tissue, the bones, connective tissue, the cartilage is also connective tissue, together forming the framework of the body. So the whole body is supported by the connective tissue. You see the blood vessel can follow the connective tissue and can divide into the capillaries so that it can the nutrition can diffuse and waste product can diffuse back to the blood. The cell that is important for the defense remain in the connective tissue and protect the tissue. Some special functions are there performed by some different types of tissues, like adipose tissue stores energy. This pigmented tissue, you can see this creates a barrier to the light. Hemopoietic tissue means the bone marrow that produces elements of the blood, like cells of the blood. Connective tissue comprises of two major components. This is the cell and extracellular matrix, means the substance in between the cell. The cells may be of two types. That cells, that resident cells, means these cells are developed in the connective tissue and remain in the connective tissue, perform the function in the connective tissue. These are the fibroblast and other blast cells. Adipocytes means it is fat cell, and some persistent stem cell means these are the parent cells that differentiates into fibroblast and other blast cells. Wandering cells, they are developed in some other area and circulates through the blood. finally settle down in the connective tissue to perform the function. These are actually visitors of that area but remain in that area and perform the function. Extracellular matrix comprises of some fibers. Three types of fibers are available: collagen, elastic, and reticular fibers, and these fibers are in the ground substance. This ground substance comprises of water, inorganic molecules, and organic molecules remaining dissolved or suspended. You see, if we see the tissue under microscope without any coloring, it is very tough to identify different components of the cell. So we stain the cell with colors. So we use two different colors. One is acidic, another is alkaline in nature. This helps in identifying the different components of the cell. You can see this nucleus we can differentiate from the cytoplasm. Cytoplasm also can containing some area which is lightly stained, some area relatively darkly stained. In this way, because of the different staining, we can differentiate the different types of cell, different components of the same cells, and so on. So we in the laboratory usually use the H and E stain, which is hematoxylin and eosin. One is acidic, another is basic. You see, with this stain, we can see the eosin stain part is reddish or pinkish in color, and the nucleus you can see the violet in color. This is hematoxylin has taken by this area. So cytoplasm become pink. Eosinophilic used to say this is affinity for eosin, and nucleus is violet or blue in color because it is affinity for the basic dye. This is basophilic. Intracellular substance in between cells are mostly stained pink, means it is eosinophilic. To describe any cell, we use three aspects. We usually evaluate three aspects. Like I have shown one large cell among other cells. This arrow, blue arrow, is indicating the cell. So how can we describe the cell? We can say what is the shape of the cell. So this is a large irregular shaped cell. We can say what is the characteristics of the cytoplasm. You look at this, cytoplasm is eosinophilic, lightly stained, containing some granules, means dotted materials. So in this way we can explain the cytoplasm. Now you can see the nucleus. Nucleus here is a rounded, lightly stained, nucleolus is prominent. In this way we can describe a cell. So we will try to describe each cell remembering these three areas. So let us start with the resident cell. Fibroblast is the most commonly available cell in the connective tissue, and it is irregular in shape with branched processes. You can see a diagram, 3D diagram, and in the lower part it is the microscopic appearance. Under microscope, this fibroblast looks like this one. So these having basophilic cytoplasm, lightly basophilic. Nucleus is usually oval and lightly stained, and this prominent nucleolus are there. It is the major contributor to the extracellular matrix, means it synthesizes fibers, collagen, elastic, reticular fibers are synthesized, and other molecules also, macromolecules are also deposited by these cells. So this is the major contributor of the extracellular matrix, and their number is the maximum. Even maybe 80% or more cells are fibroblasts. When it becomes fibrocyte, the function reduces, but still it performs the same function. Fibrocytes are relatively inactive form of fibroblast. Let us discuss the 5 features of the fibrocytes now. You can see the diagram on the left side. This cell is fibroblast, and this has become fibrocytes on the right side. You can see in the diagram nature. Other three photographs are histologically stained and shown under microscope. So in the fibrocytes, the amount of cytoplasm is reduced and the processes are reduced in number, withdrawn. So we can say this is irregular cell with a few processes. Cytoplasm is still there, and it contains decreased amount of organelles compared to the fibroblast. And if you consider the functional aspect, this is relatively less active cells but produce same thing, extracellular matrix component. These cells actually helps in repair and wear and tear. Further development is quite restricted for this type of cells. Adipose cell. Adipose cell is also known as fat cell. This is one of the resident cell, and you can see these are how does it look like in the right upper figure. These are the rounded large cells with the high power. The lower one you can see one cell. On the left side, the fat cell is a bit different. This is having fat cell is full of many unstained balloons. So these adipose cells may be of two types. One is unilocular, means having one room. Loculus means room. It's a balloon-like. But the left figure shows so many small vacuoles in the one cell. So this is multilocular. Means loculus means rooms, many rooms in one cell, and one room in one. I hope that you can identify the ring. This is special type of ring used by the king. This is looking like one fat cell, and this is known as signet ring. Signet ring has a very large top and a thin ring. So this type of appearance the fat cell shows. That's why fat cell is known as signet ring appearance. Unilocular fat cells that looks like signet ring already I have discussed. So this appearance is signet ring like, and it is usually very large and spherical shape, but if the number of cell is more, then the cell become polygonal because of the pressure from the surrounding cells. This has a thin rim of cytoplasm and a large unstained area, because hematoxylin eosin staining is water soluble, but this vacuole contains fat, so this area cannot be stained with the water soluble dyes, so remain unstained. And cytoplasm is a bit thicker in the area where the nucleus is present, which is large, oval, and or flattened. These cells are responsible for storage of energy in the form of lipid or fat. Multilocular adipocytes means fat cell, means this is a cell that contains many vacuoles of fat. So these cells are relatively smaller than the unilocular cells. They are usually polygonal in shape, and the cytoplasm contains many unstained droplets. These droplets are actually the fat droplets, so it cannot take the water soluble dyes. The nucleus is rounded and centrally placed. It is responsible for storage of energy, and the second function of this is it can release heat. That's why these type of fat cells are mostly visible in cold-blooded animals like snake or toads. but in human, at birth, we may get it by the side of the flank. Wandering cells are white blood cells of the blood and originates in the bone marrow. You can see the right side is a section of the bone showing the red stained bone marrow, and various type of cells are shown below. These cells circulates in the blood and some of them settle down in the connective tissue and perform roles there, some role. And these are, that's why known as wandering cells of the connective tissue. Let us discuss one by one the wandering cells of the connective tissue with their function. Macrophage is large size wandering cells of the connective tissue. They originate from the monocytes. Monocytes are the cells that developed in the bone marrow, and these monocytes circulate in the blood and settle down in the connective tissue as macrophages. The shape is irregular, you can see, and the cytoplasm mildly basophilic, frothy in appearance because of the engulfed substances. This frothiness or number of granules depends on the number of engulfed vesicles. Nucleus is oval and has an indentation. You can see the small rounded photographs here. It is under microscope. The nucleus is indented. But the right one is, the figure is rounded because it depends on the focus. If microscope is focused in a proper direction, the indentation only visible at one direction. Nucleoluses are very prominent. These are one of the member of mononuclear phagocytic system, means uninuclear, means one nucleus is there, but cells are responsible for phagocytosis, is present in different parts, like liver contains one type, lung contains one type. So this the series of cells are there in different parts. So connective tissue contains these macrophages as one of the representative of mononuclear phagocytic system. So it helps in defense of the tissue, so the defense of the body. The next cell, wandering cell, is the mast cell. These mast cells are the basophilic leukocytes that present in the blood. Naturally it circulates and settles down in the connective tissue, then it is known as mast cell. This characteristics, the shape of the cell is rounded or oval in shape. The cytoplasm is filled with secretory granules which are stained blue. So the cytoplasm is filled with bluish dots. You can see the high power figure. The nucleus is small, rounded, and usually occupy the central part of the cell. Here also you can see the mast cell in the figures, microscopic figures below. Especially the mast cells are present in the dermis of the skin, in the lamina propria of the respiratory tract and digestive tract. You can see the figure, the arrowhead on the right side and arrowhead and round within the circle showing the mast cell. This produces, stores and releases the chemical mediators of lymphocyte. Is another form of wandering cells present in the connective tissue. They are produced in the bone marrow and circulates in the blood, settle down in some lymphoid tissue. There they can proliferate, can increase in number, circulates again in the blood and can settle down in connective tissue to perform the function. They are with a very large nucleus compared to the cytoplasm. The cytoplasm forms a very thin rim around the nucleus. The lymphocytes may be of two types, T lymphocytes and B lymphocytes. Both are related to difference in a different. The plasma cell originates from the B lymphocytes. When B lymphocytes are stimulated, they convert into the plasma cell and can survive for less than, they die within the 3 weeks. So 15 to 20 is the lifespan of the cell when the B lymphocyte is converted into the plasma cell. They are characterized by the oval shaped cell. The cytoplasm is basophilic, and most important feature is the nuclear feature. This is clock face appearance, means the heterochromatins, means dark chromatins makes small cluster at the periphery and looks like cartwheel or face of the clock, and that's why it is known as clock face appearance. So these nuclear characteristics help the doctor to identify this cell. This cell are related to the defense. They can produce antibody and help to create a defense mechanism in the body. Extracellular matrix contains two components, ground substance and fiber. Ground substance is a fluid part, and it is colorless, viscous and amorphous, means no shape is there. The fibers are the elongated structure formed of proteins in the wall. This ground substance contains organic molecules like protein, carbohydrate, inorganic materials like mineral salts. Calcium is an important component, and all are present in the water soluble. The clinical importance of this extracellular matrix is that the fluid can be collected, accumulated in this extracellular matrix, leading to swelling. This is known as edema. So edema may be defined as accumulation of excess amount of water in the extracellular fluid. Let us see some photographs. This is, you can see the middle finger near the nail is having edema, means it's a redden area that is swollen up. So extracellular fluid means water is increased here. So it can be a very localized. You can see this is a bit larger area. The joint area is swollen because of the accumulation of the extra fluid in the extracellular matrix. So it is a bit localized, but the localized area is very larger. It can be generalized, means the whole limb is or whole body is swollen. So the fluid has come out of the body, blood, and has penetrated the extracellular matrix of the connective tissue. So the body is swollen generalized. Fibers are present in the connective tissue extracellular matrix. These are the elongated structure. The fibroblast present here polymerizes this protein to a thread-like structures. This may be collagen, elastic and reticular fibers. Reticular fibers cannot be seen under light microscope. Collagens and elastics are visible. With specific characteristics, we can identify each of them. Collagen fiber is the commonest type of fibers present in the connective tissue, and it is the key element of all the connective tissues and extremely strong. There is resistance to normal shearing and tearing forces, and composed of protein that is named as collagen, that is twisted around each other to form a very strong thread-like structure. You can see the electron microscopic appearance of the collagen fibers, and it shows has specific arrangement of the protein in it. In a stain, HE stain, it looks like this one, this pink color bundle. Let us discuss the characteristics. It is depending on on the structure, at least 14 types of collagen fibers are there in connective tissue. They are named as Roman letter 1, 2, 3, 4, 5, like this one, and it is depending gradually the number increases with a decrease in the size. So the large thickest one is the type 1 collagen fiber. This is present in the skin, in the tendon, in the bone, in the dentin of the tooth. So collagen fiber under microscope, this is left one is the cross section and the right one is the spread preparation, and this is unbranched, lightly eosinophilic thread-like structure forming bundle, and is usually wavy in nature. Better can be studied under spread preparation. Like you can see the thick bundles, pink in color, it's extending in all directions. The collagen fiber usually form the bundles, they are wavy and parallel to each other, but in some case we may get this collagen fiber forming network. As for the example, we can mention the type 4 fibers present in the basal lamella and in the basement membrane. One clinical condition is very much related to the collagen, excessive production of the collagen fiber in some scar tissue. This is known as keloid. Abnormally large amount of collagen is deposited in the scar of the skin, and that is known as keloid. You can see this scar is elevated area, has become elevated area on the surface of the skin. This is keloid. Reticular fibers are similar to the collagen fiber but extremely thin, and it matches with the collagen type 3. It forms the network in the stroma of the organ, and with spatial stain this can be visible as shown in the high power microscope here in this figure. This is spatially present in the smooth muscle around the smooth muscle cells. Hemopoietic tissue means red bone marrow. Lymphoid organ in the stroma of the lymphoid organs forming this network. Endoneurium of the neuron. The neurons is covered by a thin layer of connective tissue that is known as endoneurium. Endocrine gland stroma also contains these reticular fibers. The presence of the reticular fiber in different organs, sometimes used by the histopathologists to identify the organ and the changes in the organ. Elastic fibers are relatively less in the connective tissue, but it is spatially concentrated in some organs who are elastic in nature. These fibers composed of fibrillin and is cross-linked by the elastin and is having a very peculiar characteristics of the structure as shown in the diagram. The upper one is unstressed in unstressed condition, recoiled state, and when it is straight it looks like the molecular appearance looks like the lower figure. These are rubber-like and highly refractory and wavy in nature. They are thinner than the collagen type 1 fibers. This is present especially in the organs subjected to the stretching and bending. These are the lungs. Then elastic ligament. Ligamentum flava is the elastic. Ligamentum nuchae is the elastic ligament. Elastic cartilage contains this type of fibers, and elastic arteries, the media contains large amount of elastic type of fibers. In H stain, elastic fiber looks like this one as shown here in these three figures. They form the network of thinner wavy bundles. The middle figure, you can see very nicely the wavy bundles in the media of the arterial wall. They are dark eosinophilic and relatively thin anastomotic, unlike collagen. Collagen usually bundles and. Thank you very much for remaining.